Okay, so welcome to this next video in which we are talking about Clathrin Mediated Endocytosis and the endocytic pathway. Okay, so to revise what we've talked about so far, we have some target protein in the plasma membrane which we want to endocytose. Okay, so this target protein will have some sort of endocytic motif on its intracellular domain, which shows the cytoplasm that this protein needs to be endocytosed. Okay, what will happen is a adaptor protein complex 2 will then come and bind to this endocytic motif, and the adaptor protein complex 2s are targeted to the plasma membrane by binding to the C2b domain of the synaptotagmin 1/2 protein, and this interaction is is enhanced by the presence of phosphatidylinositol 4,5-bisphosphate in the phospholipid bilayer. Okay, right. So now what's going to happen? Well, Clathrin triskelions, a Clathrin triskelion is going to come and associate with the appendages of the adaptor protein complex 2. And what this is then going to do is it's going to polymerize, and I have now shown you how Clathrin polymerizes. It polymerized into one of these buckyball structures. So, what does this mean? What's going to happen at our membrane? So, let's say we have our plasma membrane here. So let's say we initially have this sheet of plasma membrane here. Okay, and effectively what we've now done is we put a Clathrin triskelion, shown here, underneath the membrane. Okay, we've attached it onto the adaptor protein complex 2, which is attached to the endocytic motif of our target protein. But effectively what we have done is now attached this Clathrin triskelion, shown here, to the underside of the plasma membrane. So let's colour this in purple here. Okay, so this is purple. Right, and now what's going to happen is that the Clathrin is going to start polymerizing and forming a buckyball. So, what will happen is you're going to get this Clathrin buckyball starting to form. So let's show this here. So here's this Clathrin buckyball starting to form. But the initial Clathrin protein, whoops, I'll rectify my mistake by just thickening up the line. There we go. I meant to do that. Right. Okay, so the initial Clathrin protein is down here somewhere, okay? And that's attached to the plasma membrane. So what's going to happen is the plasma membrane is going to get pulled in like so. Okay, so this is how you bud off a vesicle from the plasma membrane. So this now is known as a Clathrin-coated vesicle bud, okay? So it's coated in Clathrin, so Clathrin-coated, and it's budded off the plasma membrane, so we call it a vesicle bud. So Clathrin-coated vesicle bud. Now, at the moment, these Clathrin-coated vesicle buds, they will form without any sort of signal from the cell. And now what do I mean by that? Well, basically, you remember right back at the start of this video, I told you that when we stimulate an axon, when we fire an action potential down the axon, into the axon terminal, that action potential arriving in the axon terminal triggers endocytosis. Now, this initial step of endocytosis will occur whether an action potential is there or not. The final step of Clathrin-mediated endocytosis is what requires the action potential to be there. And the final step is the pinching off of the neck of this Clathrin-coated vesicle to actually pinch off the Clathrin-coated vesicle. Okay, sorry, the pinching off of the Clathrin-coated vesicle bud's neck, okay, so that we actually get a Clathrin-coated vesicle. Now, you might wonder, you might ask, well, if the Clathrin is polymerizing into a buckyball, why doesn't it just complete this process? Why doesn't it actually form a complete buckyball? And then the formation of that buckyball is just going to cut the membrane across there. Well, the answer is the Clathrin just doesn't have the force needed to cut the membrane up, basically, like that. It doesn't have the strength in it to actually close this neck here and actually cut off a full vesicle. So basically, as the Clathrin buckyball gets larger and larger, as it continues to grow, eventually it will stall. The polymerization will stall, and that's because the next Clathrin triskelion that you'd have to bring in just wouldn't have the force to push the membrane out of the way to latch on, basically. So it will stop, basically, when it reaches this point where it doesn't have the energy to continue. Okay, so what we now need to discuss is how do we pinch off the neck of this Clathrin-coated vesicle bud, and how is this dependent on an action potential arriving in the axon terminal. Okay, so what happens when an action potential arrives in the axon terminal here? So here's our axon terminal. Okay, now when an action potential arrives in the axon terminal, what happens is it causes the opening of voltage-gated calcium channels in the membrane of the axon terminal. And these voltage-gated calcium channels are specifically of the N or PQ type. So in the axon terminal, you only have N or PQ type voltage-gated calcium channels. So this is a VGCC, short for voltage-gated calcium channel. Okay, now when these voltage-gated calcium channels open, they're going to allow calcium to enter into the cytoplasm of the axon terminal. So in comes calcium, and we know what this calcium does. The calcium signal causes the exocytosis of the docked synaptic vesicles. So it's going to lead to exocytosis of the docked synaptic vesicles, okay? But what's it also going to lead to? The calcium also leads to the endocytosis. It also leads to the activation of this process which will pinch off the Clathrin-coated vesicle buds. So let me repeat, because it's worth repeating. These Clathrin-coated vesicle buds will form without the calcium signal. But for the next part, for the actual cutting off, the pinching off of the Clathrin-coated vesicle bud from the membrane, the narrowing of this lumen connecting the extracellular fluid to the lumen of the Clathrin-coated vesicle bud, this requires the calcium. So let's see how now. So, when calcium goes up in the cytoplasm of the axon terminal, what does it do? Well, it binds to a protein known as calmodulin. So, let's have a little bit of a discussion of calmodulin. Okay, so here's calmodulin. So this is calmodulin. Now, calmodulin has two lobes. It has an N lobe over here and a C lobe over here. And connecting these two lobes of the calmodulin protein, you have this linker polypeptide here. Now, each of these two lobes of calmodulin, the N lobe and the C lobe, has two calcium binding sites. So overall, 4 calcium ions can bind to the calmodulin protein, 2 to each of the lobes. Now, these calcium binding sites of the calmodulin protein are what are known as EFhand domains. Okay, now I want to give you some insight at least into what an EFhand domain actually is. So an EFhand domain is a type of structure that you can have within a polypeptide or a protein which is capable of binding calcium basically, and roughly its structure looks like this. Okay, so basically, if this line represents the polypeptide, so basically you have amino acid polymerized to amino acid polymerized to amino acid polymerized to amino acid, etc. It's the chain of amino acids. That's what this line represents. So basically, it forms this loop here, and the calcium ion is going to be coordinated within the center of this loop. Now, how do you coordinate the calcium in the center of that loop? Well, basically, you pick a clever choice of amino acids to put in this loop here. So what you do is you pick amino acids whose R chains are acidic. Now, why is that a good idea? Well, let me explain by giving you an example. So let's take an example of an amino acid whose R chain is acidic. So we'll take this at the amino acid aspartic acid. Okay, so here is aspartic acid. I'll firstly just draw the core amino acid structure. So here's the core amino acid structure so far, the amino group, the carboxylic acid group, the alpha carbon. And then the R group of aspartic acid is that you have a methylene group here with a carboxylic acid group coming off there. Okay, so this is aspartic acid. And the single letter amino acid code for aspartic acid is D. Okay, so that's just a little bit of added information. Right, so aspartic acid. Well, why on earth is this going to be useful in coordinating calcium? Well, the answer is it's an acid. So what's it going to do? It's going to donate protons. This hydroxyl group here, which is part of the R group, this is capable of donating protons. And you might ask, well, why isn't this one capable of donating protons? And I would say it is, but of course, this is the pure amino acid. This will be in the polypeptide in this loop. So this carboxylic acid group won't be still pure in its pure form as a carboxylic acid group. Instead, it will be involved in the peptide link to the next amino group along. Similarly, this amino group will be linked to the prior amino acid in the polypeptide. So those two functional groups won't be in their pure state anymore, so those we can ignore. This portion, however, this will still be intact. So, basically, what can happen is this hydroxyl group here, it can donate that proton that's on it. Okay, so let me show this again. So here's the carboxylic acid group drawn bigger, basically. Now, let's have a little bit more of a study of this bond between the oxygen and the hydrogen. So the oxygen gives one electron, which we'll denote as the cross. The hydrogen gives another electron, which we'll denote as the dot. Okay, now when you break this bond, how is it going to happen? Well, what could happen is you could get what's known as homolytic fission, which is where one electron goes to each of the species, okay? So in homolytic fission, and I'll write that down, so homolytic. So lytic comes from lysis, which means to break apart. Fission also means to break apart. So homolytic means that you break it and something is the same, and fission also means that you break it. So homolytic bond fission, in fact I might even have the bond in there, homolytic bond fission, means that when you break this bond, both of the species that were involved in the bond get one electron. So it's the same, they get the same out of it, so one electron goes to each. The alternative is heterolytic fission, where each one gets a different amount. So in heterolytic bond fission, what will happen is one species will get both electrons and the other will get none. Now, in homolytic bond fission, what would happen is one electron will go to the oxygen and the other electron will go to the hydrogen, and you'd create free radicals. That is not what's going to happen. Instead, you're going to get heterolytic bond fission, which is where both of the electrons are going to go to the oxygen, and this hydrogen is going to leave with nothing. I feel like I'm Robinson. Right, okay, so what happens is the hydrogen nucleus goes off with absolutely no electrons, because remember, hydrogen only had one electron, and it's lost it to oxygen. So the oxygen gets a negative charge, and the proton, as it usually is, because the hydrogen nucleus is just one proton with no neutrons. The most common isotope of hydrogen is protium, which is the isotope which has no neutrons, okay? So we'll assume we've got a protium here. Okay, so you're going to leave with just a proton. So the proton's going to get a positive charge, the oxygen's going to get a negative charge. Well, the proton already had the positive charge. The point is the hydrogen is going to go off as a positively charged species and the oxygen's going to have a negative charge. Okay, so that's going to leave this molecule now with a negative charge on, and hopefully you can start to see why this might actually be quite useful. Right, now the structure that you're left over with, the aspartic acid molecule, which has lost its proton, that has a full name. It's known as the conjugate base of aspartic acid. Okay, so the molecule of aspartic acid, once it's lost its proton, is called the conjugate base of aspartic acid. And in fact, it even has its own separate name apart from that. It's also called aspartate. Okay, so that is the strict difference between aspartic acid and aspartate. Aspartate is the aspartic acid once it's lost its proton. Now, people use them interchangeably, and that is with good reason. The reason is, if you have a liquid which contains some aspartic acid, okay, it will also contain aspartate, because basically you can't just have all of your molecules in the aspartic acid form. Some of them will be unprotonated, so some of them will be in the aspartate form, and some of them will be protonated, i.e. in the aspartic acid form. So basically, whenever you have aspartic acid, you will never have absolutely every single one of the molecules in the protonated form. Instead, you'll have a mixture of the protonated and unprotonated form. Right, so the reason it's called the conjugate base is because bases are chemical species which accept protons. So it's the exact opposite of an acid now. The aspartic acid could donate a proton. The aspartate can receive a proton. It can't donate a proton anymore, so it's not an acid. Instead, it can bind a proton onto its oxygen with this negative charge. So instead, it's a base, basically. Now, if you put lots of acidic amino acid residues in this loop of this EFhand domain here, some of them are going to donate away their protons, and some of them will keep their protons. So at any one time, you'll have a bunch of them that are unprotonated and a bunch of them that are protonated. Now, the unprotonated ones, the conjugate bases, they have a negative charge. So that's perfect then for coordinating the divalent calcium ion here. So here's our divalent calcium ion at the center of this loop of the EFhand domain. Right, now, one more thing to say is that EFhand domains rarely go around as singletons. Instead, usually what happens is in a protein, you get one EFhand domain here, followed immediately by another EFhand domain here. Okay, and this is what's known as an EF dimer. Okay, right. So, now what you've got is two of these loops next to each other, and you combine a calcium divalent cation to this one, and a calcium divalent cation to this one here. Okay, so let me colour those in orange. Now, these EF dimers are what you have in calmodulin. Each one of the lobes of calmodulin, the N and the C lobe, they both have an EF dimer, and that's why you have two calcium binding sites sitting next to each other here. Okay, and we'll continue this discussion in the next video.